There we go. A student uses this apparatus to investigate the effect of light intensity on the rate of photosynthesis in pondweed. So that's the student's method. They put a cut piece of pondweed in a beaker of water. They put a lamp 12 centimeters from the beaker. They count the number of bubbles of gas released from the cut end of the pondweed. Repeat this count for 2 more 1-minute periods. The student repeats this method, moving the lamp 2 centimeters nearer to the beaker each time, which gas is released by the plant during photosynthesis. So if you want to remember, photosynthesis is essentially the inverse of respiration. So if we saw that in respiration, oxygen was one of the reactants, we know that in photosynthesis, oxygen is a product. So the gas released by the plant in photosynthesis is going to be oxygen. Which of these is the site of photosynthesis in the plant cell? We should know that plant cells have these specialized organelles called chloroplasts, which contain chlorophyll, which are used for photosynthesis. Give one abiotic variable the student should control in the investigation. Abiotic factors can be just think of the environment, okay? Abiotic means not biotic, so not living, right? So don't say anything living. Think about temperature, pH, carbon dioxide concentration, time, right? So usually I always, when I see questions like this, the first thing that comes in my head is temperature. They usually say except temperature or something like that to make you not say temperature because it's so easy. But yeah, you could say pH, carbon dioxide concentration, whatever you want. The table shows the student's results. Plot a line graph to show the relationship between the distance of the lamp from the beaker and the number of bubbles. Okay, for stuff like a line graph, you guys can do it yourself. Like, you don't need me to show you how to do a line graph. But again, you just want to plot to the plot the line graph, right? If you want tips on how to do it, just make sure you use a ruler, right? Make sure you label your axes, right? It gives you good axes labels in the tables, so make sure you include the units as well. Okay, just plot the points they give you, and yeah, I don't know what to say. They gave you the mean already, so essentially you're basically just plotting the points and then drawing a line together with a ruler. It's not too hard. You guys can do it. So yeah. Explain the effect of increasing the distance of the lamp from the beaker on the mean number of bubbles released per minute. Okay, so we're going to just look at the data for this because we're not drawing the graph. But we can see that as we're increasing the distance, we're seeing a lower mean bubbles of number of bubbles per minute. So what is this? So this means that increasing distance, as distance increases, we see lower bubbles per minute. Okay, now explain this. So why would this be? Well, increasing distance means lower light intensity, right? Lower light intensity. Now, what does this mean? Lower light intensity means light becomes a limiting factor. Now, limiting factor is something that limits the progression of photosynthesis. So it's the thing that prevents the rate of photosynthesis from going any further. Okay, so if you have very little carbon dioxide in the room, that is a limiting factor. But in this case, we have less and less light, so it now becomes a limiting factor. And that means that we have a lower rate of photosynthesis, which means less oxygen released, which is why we're seeing less bubbles per minute. Okay. And that was it, yeah. Okay, 7. Human blood contains red blood cells and white blood cells. Give 3 differences in structure between red blood cells and white blood cells. Okay, red blood cells we know has that unique biconcave shape. Biconcave shape for RBCs, also known as erythrocytes. But for our white blood cell, right, it's not biconcave. Remember, if we're going to say one point, right, if we're writing differences, we can't just write something about one of them, right? We have to write both of them. It doesn't have to be complicated, right? In this case, I wrote biconcave shape for red blood cells, and the difference is that white blood cells aren't biconcave. It's simple as that. What other things can we say? Red blood cells have hemoglobin. We know it's that important pigment that we have that allows us to transport oxygen around our body. White blood cells, no hemoglobin. Okay. Red blood cells, remember to maximize space to carry as much oxygen as they can. They don't have any nucleus, so no nucleus. But white blood cells, it has an important nucleus. Has nucleus, right? Nucleus is very important to the function of white blood cells. So, yeah. Okay. Scientists collect data from men and women living at different altitudes. The scientists determine the mean mass of hemoglobin in 1 liter of blood and the mean mass of red blood cells in 1 liter of blood. And then these are the results. The blood volume of a woman living at an altitude of 1,890 meters is 4.3 liters. Calculate the total number of red blood cells in this person. Give your answer in standard form. Okay. So 4.3 liters is... so the blood volume is 4.3 liters, 1,890 woman, okay? And the mean number of red blood cells, times 10 to the 12, wow. Okay, so the mean red blood cells is 5.2, this one. So it's 4.3 times 5.2, 22.36, right? But remember, this forsaken unit is very large, right? It's not going to be that little. We have this unit, so we have to adjust for that. It's times 10 to the 12. But it wants us to give it in standard form. You should know that in standard form, our number should not be above. It should be a single digit number, essentially, right? Our number should be single digit. So we needed to make it 2.236 times 10 to the, and now that we've moved the decimal place here, we have to make it 10 to the 13, okay? Okay, calculate the percentage difference in mean mass of hemoglobin in 1 liter of blood in men living at 2270 meters compared with men living at sea level. Okay, so men at 2270, 151, sea level 148, okay? So the difference between them is 3, right? So it's 151 minus 158, and then we do that divided by the initial, which is 148, right? So 151 minus 148 divided by 148, and then we multiply that by 100 to get the percentage, and that gives us 2.03%, 2.03. So we see that the men at the higher altitude have a higher mean mass of hemoglobin. Not by a large percentage, but by a percentage. And if you guys wanted to know, this is how famously cyclists do blood doping, right? Some of them, they go to high altitudes, they live there for a while, and that increases their mean number of red blood cells, increases their mass of hemoglobin. Essentially, their hemoglobin also gains a property to do with their partial pressure of oxygen in there. It allows their hemoglobin to essentially pack more oxygen more efficiently, essentially, without going into the details. And they draw blood when they're up there, and then they inject that blood into their body when they're at sea level, and it gives them this advantage where they can perform better. And it's called blood doping. So you can look into that if you would like. Okay, so I want to discuss the relationship between altitude and mean mass of hemoglobin in 1 liter of blood and the number of red blood cells in 1 liter of blood in men and women. Use the data in the table and your own knowledge to answer. So from the table that we saw that just from sea level to increased altitude, from sea level to 1890, we're seeing a general increase in the mass of hemoglobin and the mean number of red blood cells. However, there seems to be a plateau effect where it's not really increasing past that at 2270 meters. So what will we write? Well, we can say that our hemoglobin increases from sea level to 1890, and then slightly decreases / plateaus, right? Yeah, we'll say slightly decreases. We'll be consistent. Okay, slightly decreases Well, this is linked to the idea as more RBCs are produced at these higher altitudes, right? So the idea is where we see more red blood cells are produced per liter, so of course we're going to have a higher mean mass of hemoglobin. But why are more red blood cells produced? Well, we have a lower partial pressure of oxygen, right? Or just the idea that we have less oxygen at these high temperatures. So we need more RBCs to sustain oxygen transport, right? Because we have less oxygen in the air, so then to spread that little oxygen that we do have in the air across the body, we need more red blood cells to facilitate that. Okay. How many? We need 5 marks. Okay. Now I want us to discuss it, so let's throw in a few limitations in the study. Okay. So different altitudes. So these seem to be. It doesn't give us any information about the number of people in the study. Oh, it does. Okay. So a lot of people at sea level, less people at altitude. So we can put a limitation. It's disproportionate. Disproportionate number of participants need more at altitude. Okay. And yeah, it's a pretty strong limitation, pretty reasonable. I'm trying to think of what else we can say. There's so many things to talk about in this data, but you can see I was quite broad in terms of how I talked about just the logical increase of hemoglobin and blah blah blah, but there's a lot of nuanced things where we see this decrease in hemoglobin, decreased mass of hemoglobin from the higher altitude, from these slight reductions, which we haven't talked about, which we could talk about. What else can we talk about? Yeah, men and women. Okay, we haven't talked about gender differences. Okay, so if you want more points, I think we've already reached 5 marks, which is why I'm not writing anything else down. But in terms of the differences, we're tending to see that men have more hemoglobin and more red blood cells at basically every altitude than women. So, yeah, it's suggesting that men are on average are having higher mean mass of hemoglobin and more red blood cells than women in general. Okay. Cystic fibrosis is a condition that affects the mucus produced in the lungs and in other organs. This condition is caused by recessive allele. State what is meant by recessive allele. This is a surprisingly difficult question because it's something that you know what it is, but how would you define a recessive allele? There's loads of ways to define it, but the one that is instantly apparent to me is an allele that is only expressed if homozygous. Right? This idea is that we need 2 of this allele for it to be expressed. Just the most easy way that I've always thought of it, but there's so many things you can say. You can say it's an allele that's not always possessed, an allele that is only expressed. That's only expressed if there's 2 copies. That is not expressed in a heterozygote, right? So many ways you can say it. Okay, B. The diagram shows a family pedigree. Some people in the family have cystic fibrosis. Okay. And then we have this pedigree chart with the key, and we want to use the pedigree to determine the genotypes of individuals A, B, and C. So we know this is a recessive disease, right? A recessive disease means that these people that are unaffected, a lot of them are likely to be carriers, right? In a family, just because they're unaffected doesn't mean they don't have that allele, right? So they might be carriers. So with the example of A, B, and C, A, B, and C. A and B, we know both A and B, they don't experience cystic, they're not sufferers, right? So we know they can't be homozygous recessive, but we know that they were able to produce an offspring that did have cystic fibrosis, right? So we know instantly that both of them, if we're using C, right? We know they both have to be heterozygous. Now, why do we know this? Well, we know this guy, C, which we also have to write C, is right? So C is affected, so we know immediately that it's homozygous recessive, right? C. So if this genotype is possible, it's only possible if the parents are both heterozygous, since both parents are unaffected. Because if you think, oh, what if one of the parents was homozygous dominant? Let's have a look. If let's say the one is dominant and the other one's homozygous recessive, we would have a homozygous dominant, a heterozygote, and a heterozygote. All of those outcomes result in the dominant allele being expressed, meaning you don't have cystic fibrosis. None of the children will have cystic fibrosis if this were the case, but it is not the case, because we have this parent being heterozygous, meaning that the Punnett square looks something more like this. And this is the one we're concerned about, because that is the one that does have cystic fibrosis. So what are the genotypes, right? It wants us to use we can use anything. Generally, you don't use letters that look similar in uppercase and lowercase. That's why I shouldn't really have used C. So let's use F, because it's CF, right? Cystic fibrosis. So A is the heterozygote. B is also a heterozygote, but C is unfortunately homozygous recessive, meaning they have the condition. Individuals E and F have a third child. Draw a genetic diagram to show the genotypes of E and F, the gametes they produce, and the possible genotypes and phenotypes of the offspring. Okay, so E and F. So E and F were able to produce a child that has the has cystic fibrosis, right? So this child is unfortunately FF. And this means that both the parents are heterozygotes. This makes life a lot easier, because the gametes they produce and the possible phenotypes. Okay, so the genotypes of the parents are this, right? The gametes they produce are that and that, right? These are the gametes they produce. Now, when they assort, they can get that, you can get that, you can get that, you can get that as their outcome. And the phenotypes are unaffected, also unaffected, affected. And if you want to know the genotypes, this is homozygous dominant, heterozygous, and this one homozygous recessive. So of course, in the actual paper, write it down neatly, but yeah, just for explanation's sake, I did it a bit messy. The gene for cystic fibrosis affects many different body systems, including the digestive system and the reproductive system. The mucus that is produced in the pancreas is much thicker and blocks the pancreatic duct. Explain the effects that this would have on human digestion. Okay, so it's saying that cystic fibrosis leads to this thick mucus in the gut, in the pancreas, and then it's blocking the pancreatic duct. So what happens if you block the pancreatic duct? Well, what do we know the pancreas produces, right? The pancreas produces amylase, a lot of enzymes, but amylase, protease, lipase. So pancreas digestive enzymes, very important digestive enzymes, be that. And then you can name them if you want. Amylase, pancreatic amylase, protease, lipase. Of course, you have to tell every amylase, right? You have other sources of amylase. You have other sources of proteases, right? But the pancreatic ones are the ones that do the bulk of digestion, right? The other sources of these enzymes are less prominent. So, what would this have on human digestion? You would see less breakdown or digestion of starch to maltose. Because remember, amylase, that's its job. It breaks down starch into maltose, right? And then you'd also see less breakdown of lipids into glycerol and fatty acids, of lipids, less breakdown of proteins to amino acids. Yeah. Cystic fibrosis can also result in the production of thick mucus which builds up in the cervix. Explain the effect that this would have on reproduction. Well, if you have a thick mucus in your cervix, it's not going to be likely that sperm is going to be able to reach your oviducts and fertilize an egg. So essentially, right, thick mucus reduces chance of pregnancy. Why? Well, less. We can say it's like an obstacle to sperm to reach oviduct or your fallopian tubes. So less chance of fertilization. Okay, students investigate the effect of mineral ions on plant growth. They use 4 solutions, A, B, C, and D. A is a complete mineral solution that contains all of the mineral ions that a plant needs to grow normally. B is a complete mineral solution without nitrate ions. C is a complete mineral solution without magnesium ions. D is a complete mineral solution without iron ions. The plant uses duckweed, which grows on the surface of water. And that's the student's method to place each of the 4 solutions in separate jars. Float 5 plants of duckweed in each jar. Use plants with the same number of leaves, same size. Cover each jar with a plastic foam. Put the jars containing the plants in sunlight. After 4 weeks, count the total number of leaves in each jar. Make a note on the size and color of leaves in each jar. Wow, what an experiment. And it wants us to state 2 variables the students kept constant in their experiment. Well, I mean, they said it. They said quite a few here, right? Same number of leaves, same size and healthy. You can say that. What else did they keep constant? It's covered with plastic film. They're in the same. Yeah, they're all they're put in sunlight, so they're all in sunlight. So the sun is fixed. Then we can say so. Number of leaves, right? Time, they all put out for the same amount of time. The exposure to sunlight, same species of plant. There's just a lot you can say here. Just say whatever you want. Explain why the students used complete mineral solution rather than distilled water to compare the effects of lacking a mineral ion. Okay, so let's see what. So they used complete mineral solution without a certain ion. And then A is the one with the complete mineral with the ion. Okay, so why use the complete mineral solution rather than distilled? Effects of lacking. Well, distilled water contains no minerals. So distilled water is distilled, right? So there's no minerals. Has no minerals. And this would be a confounding variable because instead of lacking one mineral, you're lacking all of the minerals. Yeah, and then we can say that the mineral solution contains all minerals important to growth. Right, so all the important minerals to plant growth it contains. so it should grow normally, right? And then the idea is if you have this normal control that has all the minerals, and then you have another control that has all the minerals except one, and then that plant doesn't grow anymore, you're sure that that plant isn't growing because you're missing that one mineral. Otherwise, you don't know which mineral you're missing and why the plant's not growing if you just use distilled water. Explain why the jars are kept in sunlight. Come on, like, these questions are actually... The jar is kept in sunlight, so the plants can photosynthesize. And then explain why do we photosynthesize? To produce glucose. Glucose to respire, right? So to produce glucose so we can respire. State the independent variable. So independent variable is the one that we're changing, right? What we're changing is which mineral is missing, right? The missing mineral. The missing mineral. Okay. The student recorded the total number of leaves in each jar, how the leaves are small, large, medium, and small. They recorded the leaf color and how the leaves were between... So, okay, so there's a scale for leaf color. So 0 is white and 5 is dark green. And then they have this thing, and then we see results. So you see that the most number of leaves is, yeah, of course, the control. The control has very green leaves, and then the rest seem to have quite white, pale leaves and a fewer number of leaves. So some of the observations, such as number of leaves, are quantitative, and some such as leaf size, are qualitative. State the difference between quantitative and qualitative results. Well, quantitative, it's in the name, quant, right? It uses numbers. Quantitative results are based on numbers, right? Can be measured, right? And qualitative is observed. And qualitative results are prone to biases because they are subjective. Okay. Comment on the student's results. In your own answer, you should use data from the table and your own knowledge. Okay, let's just try to go through the results bit by bit. So in the complete one, right, in the one that has every mineral, so in the control, which is this one here, the no mineral lacking, we have the most leaves and greenest plant and greenest leaves. Okay. Now, if we take out nitrates, it has least leaves, minus nitrates, least leaves, and quite pale leaves. Okay. Now, why would this be? Now, why would we observe this in the case of nitrates? But we know nitrates are important because they're used for the amino acids, the proteins in the plants. And specifically, they're also important for chlorophyll, right? Nitrates are quite important for chlorophyll, and they're used for plant growth. Now, if nitrates are important for plant growth, that quite directly explains why we're seeing that nitrates have the least leaves, because the plant isn't able to grow as much due to the absence of nitrates. So important for plant growth. For plant. Right? Which is why we're seeing the reduced plant growth in terms of less leaves in the plant that's deficient in nitrates. Okay, now let's see magnesium. So magnesium has a bit more leaves, but it also has quite pale leaves. So magnesium, we can say it has fewer leaves than control, and it has pale leaves, right? And magnesium, you should know, again, is important for chlorophyll. It's quite important in, it has an important role in producing chlorophyll. So if you get rid of it, you're going to have less chlorophyll, and that's why we're seeing these paler leaves, because chlorophyll is a pigment that dyes the leaves green. So if you have less chlorophyll, the leaves are going to be less green. So magnesium used for chlorophyll. Okay. Now, the final one is iron, right? So iron, we have the lowest leaf color. So iron, so we have less leaves, and we have we have white leaves, right? Because it said one is white, I think. Yeah. Oh no, zero is white. Um, so most white leaves or least green leaves. Okay. And then again, why is this important? Well, like all the aforementioned compounds, iron is important for the production of chlorophyll. Okay. Iron used for chlorophyll, which explains why we're seeing a pale leaf, because chlorophyll is not able to be produced and thus it's not able to dye the leaf green. Does it say discuss? Yeah, comment on the student's results. If we want to do a critique comment, we can talk about how, well, he didn't use that many plants, right? In terms of plant studies, he wanted a lot of plants. He didn't use that many plants. He was just using, these four containers and 5 duckweeds in each jar. So it's not a lot of data. It's prone to a lot of types of errors. So he should really be using more plants. Okay. Carbon dioxide can be added to a glass house to increase the yield of a crop plant. Design an investigation to find the carbon dioxide concentration used for maximum crop yield. Include experimental details in your answer and write in full sentences. Okay, we've made it to the quorums question. If you don't know what quorums is, I have a video on my channel. It's the first video on my channel, so yeah, just check out that video. It explains perfectly everything to do with quorums and how to answer it. So I'm just going to assume you already watched the video and that you know how to do quorums. We'll go right into it. So C, change, right? What are we changing? Well, we want to find the carbon dioxide concentration needed for maximum crop yield, okay? So the thing we're changing is amount of carbon dioxide. Okay? Oh, okay. Organism. How are we going to keep the organism controlled, right? How are we going to use the same? Well, we're testing a crop plant, right? So we don't want to use many types of crop plants. We want to pick one crop plant and keep the same one. So use same species of crop plant. Okay, R. Okay, we're going to repeat each concentration X number of time, yeah, whatever, right? Well, so basically, repeat each concentration is the idea. M, right? Measure. What are we going to measure? Well, we want to find crop yield. Well, what's a good measure of crop yield? The instant thing in my head is mass, right? Mass of crop you get. That can be in kilograms, I guess, if you're doing a lot of crops. If you want to be more out there, you can talk about the fruit, because some crops produce fruit, so you can talk about mass of fruit or amount of leaves. Loads of things you can say. I think mass is really easy, so mass of crops, right? And throw a unit in there just to make sure the examiner knows you know what you're talking about. It's a unit. On the second, then we want to talk about a time frame, right? Because we can't just have this forever, right? So following, you can say a specific amount of time. You could give them a suggestion. In this case, well, what time should we put? Well, following a year, right? Let's say that. No, let's say 2 months. Let's give them 2 months to grow and then see it. Okay, and then we have our S, so our controls. So what are we keeping the same? Well, if we're measuring carbon dioxide, there's many things that are important in terms of keeping the same. We know that light is important in photosynthesis, and if we change the amount of light that the plants receive, it's going to be a problem. So light availability, right? What else? Well, we can talk about temperature, right? I wouldn't say that's the best one because, I mean, it's a pretty good one, honestly. Yeah, temperatures, I don't know what I'm talking about. Temperature's a good one. I was going to say that temperature would be a bit hard to control, but in this case, if you just keep all the plants in the glass house, it's not going to be a problem. What else can you say? You can talk about the soil that the plants are in. You can talk about doing them at the same time, because let's say you have some of the plants in winter and then some of the plants in summer. It's going to be quite completely different, right? And that links into the temperature as well and the climate. But again, the season would be a nice thing to say. You can talk about the minerals in the soil, the pH of the soil, the presence of pests, right? That's something you can say. So many things you can say. Yeah. Okay. And there we go. So that should be 6 out of 6. That should be the last question. So yeah, we've got through that paper relatively quickly. It wasn't too bad. At times I was a bit rushed because I do have to go to sleep soon. Have an early morning lecture tomorrow. But yeah, hopefully you guys enjoyed that. If it was helpful, please let me know in the comments, and let me know whatever other papers you'd like to do. I do try and actually, this paper was recommended to me by a subscriber. So let me just find the name. Yusuf Khan 5715. Thank you for suggesting this paper. It's helpful because I don't really know what paper you guys like me to do, so it's helpful when I get suggestions like that. But yeah, if you want me to do any papers in specific, just suggest me the paper. I'll do it as quickly as possible. Hopefully you like this. And yeah, show some love to the channel, guys. It's really appreciate you guys. Yeah, see you in the next one.